This is a demonstration of Unitech's FrSO2 assay on the EnoLyzer. We will be testing 2 wine samples for FrSO2 as part of a second part of our demonstration of testing both malic acid and FrSO2 on the EnoLyzer. Refer to Unitech's malic acid YouTube video. As a part of this demo, we will also show the use of StatFacts capture software on our PC to create an Excel file of our EnoLyzer data. Now, the FrSO2 assay on EnoLyzer. Refer to EnoLyzer application sheet. The same wine samples used for malic acid testing will be tested for FrSO2. Sample prep is the same, though centrifugation is required today. FrSO2 testing requires the use of 2 sets of tubes for each blank, standard, and sample. Results from the first working blank set is used to subtract the sample color from our calculations. EnoLyzer makes this sample blanking procedure easy. The first step, remove the reagent vials from the Uniflex FrSO2 reagent kit. We have diluent, oxidizer, chromogen concentrate, starter, and our 20 milligram per liter sulfite standard. We're planning 1 ml of working reagent for each tube. The tubes include blank, standard, a control, a red wine, and a white wine. Today we're going to run a 50 milligram per liter sulfite standard as a known sample or as a control to check the performance of our assay. If desired, the volumes from the EnoLyzer application sheet could be scaled downward, keeping the same ratios, in order to save reagents. Today we will pipette as shown for 4 tests. Step 2, preparing FrSO2 working blank and working reagent. Label the bottles as above, working blank and working reagent. Select the AP 100 to 1000 pipetter. Set the pipette at 1000, that's 1000 microliters. Use a blue pipette tip. We'll be pipetting 5 times to deliver 5 mils of diluent into the working blank bottle, as shown on the table in the EnoLyzer application. Carefully aspirate and dispense 5 times into the reagent bottle for working blank. When we have 5 mils of diluent in the working blank, we'll repeat that 5 mils of diluent into the working reagent bottle. The next step is to pipette the oxidizer only into the working blank bottle. We'll select the AP 200 to 20 to 200 pipette and the yellow tip. Set the pipetter at 50 to dispense 50 microliters or 0.05 mils of oxidizer. We aspirate the oxidizer, carefully pipette it into the working blank bottle and mix. We now select the AP 100 to 1000 pipetter again, set it at 500 microliters to pipette 0.5 mils. This requires a blue pipette tip. We're going to aspirate 500 microliters of chromogen and pipette it into both working blank, and then aspirate another, pipette it into the working reagent bottle. With a new blue pipette tip, we will aspirate 0.5 mils of starter and pipette that into the working blank, repeating that step, 0.5 mils of starter pipetted into the working reagent bottle. The third step is to label our reaction tubes. We have the tubes set up in our blue test tube rack. We're labeling 2 sets of tubes as blank, standard, control, white wine, and red wine. The first set will receive working blank and the second set will receive working reagent. Here's an overview of our procedure. We're pipetting 25 microliters of distilled water into the blank tubes, now 25 microliters of standard into the appropriately labeled tubes, then 25 microliters of control, then 25 microliters of white wine into both the blank tube set and the reaction tube set. And finally, we'll be dispensing the red wine, 25 microliters of red wine into each red wine tube, the blank tube, and the reaction set of tubes. With the samples dispensed, we'll go back to use our AP 100 to 1000 pipetter and the blue tip. First dispensing the working blank into the blank set of tubes on the left. So we're setting the pipetter at 1000 for 1000 microliters to pipette to 1 mil or 1000 microliters of working blank. By keeping the pipette tip above the reaction solution, we don't have to change the tip between samples because we're not contaminating the tip with sample. So the working blank tubes are ready to react. We will now change the pipette tip, get a new blue pipette tip, and dispense the working reagent into the 5 working reagent tubes. Notice that these tubes are being incubated at room temperature in the blue rack. We're not using the temperature controlled incubator on the EnoLyzer. For free sulfite, room temperature is preferred to 37-degree incubation. Now, following our EnoLyzer application sheet, we turn on the EnoLyzer. Well, we're setting the timer for 5 minutes. Now we turn on the EnoLyzer. We connect the printer cable to the back of the EnoLyzer. The other end is connected, the USB end is connected to our PC. We open up the StatFax Capture software on our PC. And on the EnoLyzer screen, we will, on the startup menu, select Run Test. Our FrSO2 assay is test number 10, so we will have to go to the next page, double arrow to the right, and then select test 10. Yes. Notice on the PC that it has placed the name of our assay at the top of the sheet. Now when the buzzer goes off, we will continue with our assay by reading the tubes. The EnoLyzer is prompting us print full header. We say yes. We'll have a full header today. It shows us the previous free sulfite curve we had run. We're going to discard that because it's been more than 3 hours since we ran our free sulfite test, and we'll be running a new curve today. It asks the number of samples. We count samples including the control, so that will be 3. The buzzer has gone off, so we're going to enter the blank tubes. First, we'll enter the working blank set. The blank tube is reading. We remove the tube. Insert standard. Remove tube. Insert sample 1. That's our control. Insert sample 2. That's our white wine. Insert sample 3, that's the red wine. So all of the initial absorbances or the blank readings of absorbance for the samples have been completed. EnoLyzer prompts us insert blank. It's talking about the reaction set of tubes. The DI water blank tube is inserted. Remove tube. Insert standard. Remove tube. It shows us the graph of the blank and standard 2-point curve. The absorbances look proper to us. We accept the curve. Insert sample 1, that's our control. Remove. Insert sample 2, that's the white wine. And insert sample 3, that's the red wine. The results are shown on screen. They're also printed out on the printer tape, and you'll notice on the PC that the results have all been captured on the StatFax capture software. Our control came in at 50, and this was our 50 control. So we're happy with our assay performance. And that concludes the FrSO2 demonstration of the EnoLyzer.